up to 10 to 70% large, but when the variety is susceptible, it can cause up to 100% large. It reduces the grain yield, nutrition, and quality of the crop. It can be controlled by chemicals, but chemicals has its own limitations because it is expensive, cannot be afforded by the farmers, and also it's non-environmental friendly. So the only suitable option is its control through genetic resistance. So there are two more types of resistance. One is the major gene-based resistance, which is non-durable, and minor gene-based resistance, which is durable. But how we can bring this type of resistance in varieties? So we have to explore diverse sources of resistance. We have to look for both indigenous and exotic germplasm. So if this is a durable approach, so for this we have to so the genetic resistance is actually overcome by the activation of virulence by the pathogen, and that would be explained by the boom and bust cycle. So what is the boom and bust cycle? On the left side, you can see that when a breeder produces a new variety, its farmer likes it and its area increases. So that is the boom of the variety. And the pathogen population is decreased, so that is the bust of the pathogen. But when a new mutant is detected, its frequency increases and it causes epidemic. So that is the boom of the pathogen when the area of the variety is decreased. So this is the bust of the variety. However, it requires knowledge of the resistance status of the varieties, and also population structure of the pathogen would be considered. Now, from where this pathogen originated? So it was previously believed to be originated in the Transcaucasia, where grasses were the primary host, and from there the pathogen spread to East Asia and North America, and West Asia and East Africa. But there was no scientific reasons, it was only based on evolution. But the very recent study of Alita 2014, it was identified that Himalayas and near Himalayas are the center of origin of the pathogen. And their conclusion was based on pathogen population structure, center of diversity, high sexual reproduction rate, and the prevalence of its alternate host. So this is the worldwide presence of the pathogen. It originated in the Himalayas and near Himalayan region, and from there it spread to other parts of the world, and to some regions from the secondary sources. Like you can see that it moved, it spread from the center of diversity to the Mediterranean region, and from there it spread to Australia and America, as you can see by the red arrows. So what is its population structure in Pakistan? The worldwide study identified the Himalayan and near Himalayan regions to be the center of diversity. So it has an overall high diversity in Pakistan and sexual recombination in the northern region of the country, while it has relatively low diversity in other regions. So information on the Pak-Afghan bordering into Bush area is limited, and this area lies in the ex-tribal districts that were not accessible mainly due to security situations. So we took advantage of the conditions of the past few years, and we organized this study. The objectives of this study are to evaluate the status of weed diseases across different locations in weed-growing regions of the newly merged districts of Pakistan. The second objective is to describe the level of diversity and recombination in the pathogen population. And the third objective is to assess the temporal genetic shifts over years. And the fourth objective is to assess the relationship of ex-tribal district population with the worldwide areas. Material and methods. So the first experiment was the distribution of status of various weed diseases. So this is the map. In the map, you can see that we have surveyed different districts over the 4-year surveillance made during 2019 to 2022. And you can see it by the red dots. It shows the districts that we have covered over the 4 years. This is the pictorial details of the surveyed district. We covered North Waziristan, South Waziristan, Pahad, Mumbai, Major, etc. So what we did in the field, we scored the disease based on two parameters. One is the host reaction and another one is the disease severity. So host reaction actually ranges from immune to resistant to moderately susceptible, and then susceptible. So if you have no infection, that could be considered as immune. If you have necrotic areas without pustules, with or without pustules, it could be regarded as resistance. And if you have pustules plus necrosis, then it could be regarded as moderately resistant. And if pustules plus fluorosis are present, it could be regarded as moderately susceptible. And if you have large pustules with no necrosis, it would be considered as susceptible. And disease severity was, for disease severity, we used modified Cobb scale, which is based on the percentage of the area infected and the overall blood infestation. And we also calculated the coefficient of infection based on disease severity and host reaction values. Now this is experiment number 2. So for experiment number 2 was actually based on population structure. So for that we collected samples of the pathogen. We actually collected leaf samples from fields, and these were placed in lysine bags and were labeled properly for future use. After that, we isolated lesion using sterilized layers, and each lesion were put in a build-up tube for further processing. For molecular genotyping, we first extracted the DNA using modified CTAB method, and that DNA was amplified in 2 PCR multiplexes using 17 SSR markers, and PCR products were run on jet for confirmation, and then the PCR products were sent to China for sequencing. So these are the details of the SSRs that were used in the 2 PCR multiplexes. These are the output chromatograms that were generated by the sequencer, and we read it through GenMarker software to score alleles, and the scored alleles were formatted in input file for further they were used in population softwares for further analysis. So for population genetics, we use different softwares. The first one was genetics that was used to estimate the allele frequency observed and expected heterozygosity. FST multilocus was used for detection of multilocus genotypes. For genotypic diversity, linkage disequilibrium among the marker pairs. GeneClone was used for resampling of multilocus genotypes and plotting of multilocus genotypes against the number of loci. And population was used for phylogenetic tree. A degenerate package of R software was used for DAPC analysis, and FSTAT was used for allele richness in gene diversity. And the data was analyzed based on region, district, and host varieties. Now, experiment number 3 was to assess the temporal genetic shift of the pathogen population over the 2 years. For that we assessed FST values between the years, and we also did resampling of the multilocus genotypes over the 2 years. And then we estimate sexual reproduction rate and effective population size by using clone case method. So what is the base of clone case method? Let's assume you are doing sampling at time 1, that is sample 1, and you have different genotypes, and then you are doing sampling at time 2, that is sample 2. Again, you have different genotypes. So for yellow rust, we know that a part of the population is passing through sexual reproduction, while a part of the population is passing through clonal reproduction. So, based on the resampling of clones, you can estimate S, which is the ratio of sexual reproduction, and 1 minus S, which is the ratio of clonal reproduction. So the more you have the clones, the more the clonal population, and the less the clones you have, the less the clonal population. Now this is experiment number 4, which was actually based on the comparison of ex-tribal districts, the senior tribe boundaries population with worldwide lineages. So for that, we used microsatellite data for worldwide lineages, and that data was combined in quality check with the representative images from the ex-tribal districts. Now results. The first experiment was the status of various diseases. So you can see here the incidence of ear rust across different regions. So the disease was found with the maximum frequency in central KP, X Sparta and X Sparta, while it was found with minimum frequency in south KP. This is the yellow severity and coefficient of infection. In the upper graph, you can see that the severity was found much higher for X Sparta during 2019 and 2020, while it was found with low severity in 2021 and 2022. Also, the coefficient of infection was found the maximum for X Sparta and during 2019 and 2020, while it was found lower for 2021 and 22. This is the incidence of yellow rust across different districts. So you can see here that most of the districts were found with the maximum frequency during 2019 and 2020, while the disease was found with low frequency in most of the districts, while with high frequency in a few of the districts during 2021 and 22. This is the mean severity as observed across different districts. So the disease was found with the maximum severity in most districts of the X Sparta and X Sparta, while it was found with low severity in South KP. This is the coefficient of infection is regarded for different varieties. So you can see here that the maximum was regarded for Galaxy, that is 83.6. While the minimum was recorded for Rebecca, that is 1.9. So most of the varieties has low overall is ACI. The incidence of reflux is some sporadic appearance in X Sparta, and central KP. This is the severity of leaf rust. So you can see here that for South KP it was found the severity was found the maximum for the South KP during 2020 and 2022. And again, the coefficient of infection was found the maximum for South KP all the years, while it was found the minimum for other regions. This is the incidence of leaf rust across different districts. So again, you can see here that the maximum districts were the maximum frequency were found for South KP, as most of the districts were found with high disease, like in Bannu, Khan, like Himarwat and Tan, while it was found with low frequency in the other regions such as Central KP, X Sparta and X Sparta. This is the mean severity of leaf rust across different districts. So the severity was found the maximum for South KP, while it was found with the minimum in the other three regions. There is the coefficient of infection of leaf rust is regarded for different varieties. So the average coefficient of infection ranged from 0.0 to 7.3, where the maximum was found for landrace. So leaf rust was found absent in most of the surveyed varieties, but still present with some sporadic appearance on some varieties, like Pirsabak 2005, Pirsabak 2013, and Shaka. It shows deployment of the varieties in regions with disease, with favorable environment for the onset of the disease. There is the incidence of powdery mildew observed for different regions. So you can see here that the powdery mildew was found with high frequency in X Sparta and X Sparta, while it was found with low frequency in South KP and Central KP. This is the severity of powdery mildew and its coefficient of infection. So the graph shows the severity. So the severity was found the maximum for X Sparta all the years, while it was found the minimum for other districts. And the coefficient of infection was also found the maximum for X Sparta, while it was found the minimum for Central and South KP. There is a powdery mildew incidence across different districts, so you can see the more the red you have, the the more the frequency of the disease in these districts. So you can see that most of the districts were found with high frequency in ex-Sparta, while it was found with low frequency in South Cape. This is the mean powdery mildew severity as observed across different districts. So again, you can see here that the maximum severity was found in most of the districts of ex-Sparta, while it was found with the least frequency in South Cape, while the severity was found in a few districts of Central Cape and ex-Sparta. There is a coefficient of infection for the surveyed varieties. So the average coefficient of infection ranged from 0 to 13. The maximum was recorded for us 2011, while the minimum was recorded for most of the varieties. This is the summary of experiment number 1. So yellow rust was the most important disease of wheat in the studied region. Powdery mildew was the second most important, and leaf rust was in low incidence, while stem rust was absent. So it will be interesting to assess the population structure of the yellow rust pathogen. Now experiment number 2, that is what we have done, we assess the population structure of yellow rust pathogen. So for population structure, first this table shows the summary statistics of the markers that we use for population structure. And you can see that all of the markers are polymorphic with the allele number ranges from 1 to 11. So a high polymorphism is detected in the markers, while the only one marker was monomorphic, that is RJN9. So to check whether the SSRs are suitable for the study or not, so we plotted it against the number of loci. So you can see here that when the number of loci increases, the number of multilocus genotypes also increases. So while reaching 10 to 11 or 12 markers, we reach up to the maximum level. So these are, we have used 17 SSR markers in our study, so these are more than enough for the study. This is the linkage disequilibrium that is detected among the loci. So apart from here, the red color shows the more the linkage disequilibrium, while the blue color shows the less linkage disequilibrium. So apart from RJO27 with RJN8 and RJN11 with RJN3, all of the markers showed low overall linkage disequilibrium. So all of the markers are so you cannot exclude any of the markers from the study. All of them are equally important. So what we did, we assessed the population structure first across the geographical regions. So the first thing here is to see is diversity. So you can see that the diversity was very high in all the three regions. There is the northern Hindu Kush, eastern Hindu Kush, and southern Hindu Kush. And the diversity ranges from 88% to some 96 or 97%. So there is a huge diversity that was observed in the regions and over years. The second, we look for heterozygosity. So the worldwide reference in the left bottom, you can see that when you have a clonal population, you will have a high observed heterozygosity. But if you look to our population, there is not a lot of difference between the observed and expected heterozygosity. So it's pointing towards the combination in the regions. Then we look for population subdivision. So you have principal component analysis in A, principal coordinate analysis in B, and phylogenetic tree in C, and network analysis in D. And the different colors is depicted by the different populations, and their distribution is actually according to their genetic background. So you can see here that all of the populations are mixed, and there is no population subdivision or no divergence due to geographical regions. The divergence of the population subdivision was further confirmed by the divergence parameter, that is FST. So what is FST? Basically its value ranges from 0 to 1. So near to 0 means no divergence. So you can see that most of our values are close to 0. It's also it also suggests the lack of any population subdivision. Then we look for multi-locus genotypes distribution. So you can see here that the MLG 166 was resampled 34 times. So the question was, if you have geographical structuring, you will have one MLG specific to a particular region. But here you can see that none of the MLG is specific to a particular region. These MLGs are distributed across the regions and across the and also across the years. Then we look for population structure across the districts. So the first thing again here is to see is diversity. So you can see that the diversity was observed was very high, and it ranges from some 72% to 96-97% for all the districts over the 2 years. Then we look for heterozygosity. So again, you can see the worldwide difference. When you have clonal population, you will have a high observed heterozygosity, while for recombinant population, there is not a lot of difference. So you can see to all populations that are very similar and differences, like you can see in the worldwide difference. So the difference between observed and expected heterozygosity was found very low. So it's also pointing towards the combination in the region, and it also reflects on high diversity. So for different districts, across the districts, we look for population subdivision, whether there is some divergence or not. So again, you can see you have principal component analysis in A, principal coordinate analysis in B, phylogenetic tree in C, and network analysis in D. And again, you can see that all of the populations are mixed, and there is no structure due to districts. This population subdivision was further confirmed by the multi-locus genotypes distribution. So you can see here that all of the resampled MLGs are multi-locus genotypes. They are distributed across the districts, like the MLG 166 was resampled 6 times in Bajod, 8 times in Munir, and 3 times in Malakand, and once in North Waziristan, and so on. So this also suggests the lack of any district-dependent structure. The divergence was further confirmed by the FST. So you can see here that all of the values are close to 0. So it also suggests the lack of any district-dependent structure. Then we look for host genotypes, the population structure across the host genotypes. So again, the first thing is to see is diversity. So on different hosts, you can see that the pathogen populations were quite diverse, and the diversity ranges from some 66 to 98%. So there is a huge diversity here for observed on different host genotypes. Then we look for heterozygosity. So the observed and expected heterozygosity, the difference between observed and expected heterozygosity for different hosts was found very low. So it's also pointing towards recombination in the pathogen populations. Then we look for population subdivision for the host genotypes. Here you can see that all of the pathogens therefore isolated from different hosts. All are overlapping, and all are mixed. So there is no host. This also suggests the lack of any host-dependent structure. Then we look for resampling of multi-locus genotypes detected in the pathogen populations. So you can see that there is not a single MLG specific for a specific host. All of the multi-locus genotypes are distributed across the host genotypes. So this also suggests the lack of any host-dependent structure. Then we look for divergence in terms of FST values. So again, you can see the FST values are very much close to 0. So this also suggests the lack of any host-dependent structure. So what we did without considering the geography and host, we did another kind of analysis. And in this kind of analysis, the software will identify the genetic groups. So in our case, these are the 6 genetic groups. And then it will make a scatter plot which will show you how these genetic groups are distributed. So you can see that group 4 and 5 are very much diverged, and group 1, 3, and 6, they are the related ones. And also you can see in the graph C that these genetic groups are not specific to a specific region and not specific to a specific year. But you can see that all the genetic groups are distributed across the districts and across the regions. This also shows the distribution of genetic groups across different regions. So you can see that all the genetic groups are distributed in all the regions. But the group number 6 that is represented by the black color is the most prevalent one in the 2019, while it was found with low frequency in 2020. This is the distribution of the genetic groups on different hosts. So there is no group specific to a specific host. All of the groups are resampled on different genotypes. And on one genotype, more than one genetic group was identified. So like you can see for the unknown genotypes in the galaxy, all of the six groups were identified. So you have, if you have the more sample size, there is a chance that the more genetic groups will be identified on the varieties. So summarizing experiment number 2, Yellow pathogen is highly diverse in the studied area. There was no spatial structure due to geographical regions, due to districts, due to host genotypes. So this reflects on gene flow across the regions and over different hosts. So it means that in epidemic in one area, a host will also affect the other area and host. Now coming towards experiment number 3, we assess the temporal genetic shifts in the pathogen populations over the 2 years. So again, the first thing here is to see is diversity. So over the 2 years, similar level of diversity was observed, like 95 and 98 is very much close to each other. So the diversity did not change over years. Then we look for the observed and expected heterozygosity over the 2 years. So you can see that the difference between observed and expected heterozygosity is very low, while it is much reduced in 2020. So this shows that the pathogen is passing through sexual reproduction, and it also reflects on high diversity. Then we look for population subdivision over the 2 years. Again, you have principal component analysis in A and principal coordinate analysis in B, phylogenetic GNC and network analysis in D. And you can see that both the populations are mixed, except there is a slight shift between the two populations of 2019 and 2020. But we cannot clearly say that there is a population subdivision between the two populations. This, the population structure is evolved over the 2 years was further confirmed on the basis of multi-locus genotypes we sampled over the 2 years on different hosts. So you can see that all of the genotypes were attacked by multiple images over the 2 years. So it also shows the lack of any year-specific structure for different hosts. Then we look for the sampling of multi-locus genotypes over the 2 years. So you can see in the upper graph that most of the multi-locus genotypes are sampled once they were not resampled. But a subset of multi-locus genotypes that were resampled, like in the lower graph, you can see that these are the multi-locus genotypes that were resampled at least twice. Like in the bar, the both colors, the red and blue, shows that the MHG were resampled over the both years. Otherwise, the only red and only blue shows the resampling of multi-locus genotypes. genotypes over 1 year only. So based on the information on resampling of multi-locus genotypes, we estimated the effective population size and sexual reproduction rate. So here in one means one clonal cycle before the onset of sexual reproduction and one clonal cycle after the onset of sexual reproduction. But this is something actually you cannot directly estimate, but you can assume that for that if you have more clonal cycle, like if you have 1 to 10 cycles before the onset of sexual reproduction and 1 to 10 cycles after the onset of sexual reproduction. But this actually does not affect on the estimation of sexual reproduction rate. As you can see here, that in all of the cases, 92% of sexual reproduction is estimated, and like about 7.8% of the population is passing through clonal reproduction. And also the effective population size is very high than the sample size, which also reflects on diversity. Then we look for multi-locus genotypes across the regions. So you can see that in Eastern Hindu Kush, none of the MLG was resampled over the 2 years. While in Northern Hindu Kush, some few MLGs were resampled over the 2 years. While in Southern Hindu Kush, there is only one MLG that was resampled over the 2 years. So this information was, the information on MLG was used for the estimation of effective population size and sexual reproduction. So here you can see that the maximum sexual reproduction was found in the Eastern Hindu Kush, that is 100%, while 72% of sexual reproduction was found in Northern Hindu Kush and 92% in Southern Hindu Kush, while the effective population size is higher, was found the maximum for the Northern Hindu Kush, while in Southern and Eastern Hindu Kush it ranges from some 379 to 495 and up to 2000 and onward. This is the summary of the experiment number 3. So no strong shift between the two populations was observed. A high proportion of the population is passing through sexual recombination, and even in each geographical region, the recombination rate was high. And the different hosts were attacked by multiple immunities over both the years. Now, experiment number 4. In this experiment, we compared the Yersinia striatalvis populations from the Strait region with the worldwide spread lineages. So, for comparison with worldwide populations, we retrieved the data from populations of Europe, China, Nepal, Bhutan, Middle East, and Mediterranean regions. And the data was retrieved from the articles of Ali et al. 2014, Class Pathogens, Ali et al. 2017, Frontiers in Plant Sciences, Ali et al. 2018, Plant Pathology, Khan et al. 2019, Phytopathology. And different diversity parameters were compared with the current data set. So you can see on the left side is our current data set, while on the right side is the different populations of the world. So you can see that in our population, the diversity was found higher than the population of Europe. You can see that in our population, the diversity ranges from 72 to some 96 or 97%. But this diversity is higher than the European, South African, Mediterranean, and Middle East population. Like the diversity is 35, 33, and 43 and 45%. While the diversity was lower as compared to the population of China, like in China, it was previously identified about 99% diversity, while a district of Nepal, where 100% of the populations were found diverse. And also a district in China, having very high diversity, which is higher than in our population. And also in 2002 to 2005 in China, the diversity was found some 99%, which is also higher than our populations. So we also compare the populations with other Pakistani populations. So the data was retrieved from different articles from 2014 molecular ecology, mycologia, versatile fungal genetics and biology, and we also compared the different diversity parameters with the current data set. So you can see in the on the left side, this is our population, so the diversity range from 72 to 96%. And this diversity is lower than the diversity as observed in Tabad, Mansehra, Patagram, Balakot, and Skardu, and Banaval. Not so again in 2011 in Button, Patagram, and Balakot. So this is lower than the diversity as was observed in the Hazara divisions in Gilgit-Baltistan. Again, you can see that in Gilgit-Baltistan, 97% diversity was observed in the previous years, while in case of our population, it ranges from 72 to 96%, so it is up, but the diversity observed here is the same level like that was observed in Khyber, 97%, Peshawar, 93%, Haripur, 67%. So the diversity was the similar level with the other Pakistani populations. So, summarizing experiment number 4, the population from the studied area were more diverse than the global population of Europe, USA, and Australia, and it was equally diverse as the population of East Africa, South Asia, and Bhutan, while it was less diverse than China and Nepal. Within Pakistan, the population was equally diverse as the population of Punjab and JP, while less diverse than Skardu, Balakot, Tarshish, Patagram, and Gilgit-Baltistan. Now coming towards conclusion and recommendations. So first, for disease surveillance, we concluded that yellow rust was found with high disease, and this is the main disease of ex-tribal districts, Central and South KP. In Central KP, ex-tribal districts, these areas were more prone to yellow rust, while South KP was more prone to leaf rust, while powdery mildew was found with high frequency in the ex-Sparta regions. And yellow rust pressure was high in 2020, leaf rust in 2022, and powdery mildew in 2021. So it is recommended that breeding programs in the area should focus on yellow rust, leaf rust, and powdery mildew resistance. Yellow rust resistant varieties should be applied in the whole KP, while leaf rust resistant varieties should be applied in the South KP, and powdery mildew resistant varieties should be applied in ex-Sparta. So cultivation of susceptible varieties should be stopped, in new and resistant varieties should be adopted. And for population structure, we concluded that population subdivision cannot be explained by geographical regions and host varieties. There was no population structure across districts. The population were more diverse than the populations of Europe, America, and USA, while equally diverse is the population of Himalayan region. And within Pakistan, the populations were less diverse than Gilgit-Baltistan and Hazara division, while equally diverse is the populations of other regions. So it's recommended that diverse sources of genetic resistance should be applied considering the diversity in the pathogen population, and regular tracking of the population should be done considering the potential aggression over the region, and breeding varieties must be done at the center of diversity and area with more disease onset. For temporal maintenance, we concluded that there was no strong shift over the 2 years between the two populations, and this is due to a role of sexual reproduction and high effective population size, and high diversity was observed over the 2 years, and the global maintenance was present but to a limited extent. So it is recommended that the role of Barbarian sexual reproduction must be considered in development of resistant varieties. The breeding effort must be continued as the diversity does not decline over time, and varieties should be replaced after every 3 to 4 years, and the role of sexual recombination in generating novel variation should be considered. And the clonal turnover during the years reflect on the possibility of survival of best fit lineages, so it should be considered by developing resistant varieties. Now, for comparison with worldwide population, we concluded that a higher diversity was observed in the global population of Europe, USA, and Australia, and same level of diversity as in other Himalayan and near Himalayan regions. And the Hindu Kush region had high proportion of the population passing through sexual reproduction, and this is also in line with the wide prevalence of its cost in the region. So it's recommended that the region could serve as a source of new variants to other regions of Pakistan, and breeding material should be tested in these areas, and the surveillance should be continued through other neighboring regions in the context of invasion, and collaboration required at different global level to explore and the collaboration required at global level to explore the population structure in the region. So these are the achievements, the meetings, workshops, and conferences that we attended in 2009, September 2010. Submitted our abstract, and that was accepted for post competition, and it was later on published in the proceedings. In 15 to 17 September 2022, we attended the fourth international conference on emerging trends in bioinformatics and biosciences, and the abstract was accepted for oral presentation, and we presented our work over there, and work was later on published in the proceedings, and this is a long list. So, and these are the four consecutive meetings in 27 to 28 September 2021. It was a national youth planning meeting at WRI, ARI, Faisalabad, Pakistan, and we attended that meeting on the 22nd and 23rd September 2020. It was an annual youth planning meeting in Islamabad, and we also attended that. And also on 24th January, it was also in Islamabad, so we attended that meeting. So these are the publications that we published so far, and the first one is from my PhD work, and the other five are from the collaboration work with my colleagues, with my friends. So the first one we have submitted from our work to ACS Omega with an impact factor 4.1. So we hope that they will accept it. Acknowledgement. So I'm much thankful to my PhD supervisor, Dr. Sajid Ali, who supported me, guided me, and he bring me on the track to get done. I would like to thank CEMET USDA Pakistan, Dr. Muhammad Imtiaz and GP Diwali, who assist our PhD work, particularly the surveillance part. I would like to thank GRRC, RSC University Denmark, and NAQUIST and NFU University China for their collaboration and sequencing. RFG faculty, RFG for particularly genomics and bioinformatics lab and biochemistry lab, especially Professor Dr. Sajan, Professor Dr. Iqbal Munir, Professor Dr. Akib, Professor Dr. Muhammad Sayed Khan, Dr. Sayed Qazi, Dr. Shehya, Dr. Ibrahim Sah, and all the staff members. Also, I would like to thank the research students who collaborated us in this research work, especially Mohammad Ramis Khan, Amir Iqbal, Safi Shinwari, Amir Mohammad Khan, Hyder Khan, Mohammad Arif Shahidullah Khan, for the field staff. And finally, I would like to thank those who facilitated our surveillance work, Dr. Atifur Rahman, Dr. Muhammad Shafi, Dr. Khalid, Dr. Gulzar, and Dr. Bashir, Dr. Fazludin Sultan, Dr. Sajid Safiullah, Khilwat Afridi, Munsifur Rahman, and Dr. Sheikh Nawab and Dr. Muhammad Awais. And thank you all. If you have any questions, remarks, or comments.
Questions, comments? Yeah. First of all, very well done. You did a very good. Simple question है कि clarification ये जो आपने years लिखे हैं 2019, 20, 21, 22. So are these in 2019, 20? मतलब क्या growing season क्या है वो मुझे समझ नहीं इस चीज़ की.
19 में भी किए, 20 में भी किए, 21.
लेकिन बीज जो है ना वो grow किया जाता है in 1 year.
Yes.
And harvest in the next year. तो क्या है आप सभी ये डेटा का टाइम है या ये
मार्च से लेकर मई तक हम डेटा कलेक्ट करते हैं।
तो इसको आपने लिखना है 2019-20 2020-20 ठीक है, क्योंकि ये तो क्योंकि अच्छा आपका जो डेटा में जो 2022 में मैंने कहा था कि येलो रस्ट और इनकी चीज़ों के इंसिडेंट बहुत कम है, हालांकि लास्ट ईयर मतलब 2022-23 में बहुत हाई इंसिडेंस रहा है, तो इसलिए मैं ये क्लियर करना था। चलो इसलिए दूसरा ये बताया कि आपने 4 मुख्तलिफ डिजीज से जो 3 राष्ट्रीय और एक में कोविड, तो हमारा एपी का is it the first report that heat first is the most important or highly prevalent or has someone is there as well?
Yes, sir. مخكدي بنكاشر الرميز شكون مخكدي identify challenges with the dry day or weather conditions that the temperatures से वादी नहीं होती हैं। जो first ज़्यादा temperature का मुझे
Sorry, yellow rust, not heat first. Yellow rust is the most prevalent in KP.
Yes, sir.
दूसरा ये बताया आपने आपका जो PCA मैंने देखा था कि इसमें अगर आप देखें दोनों कंपोनेंट 1 और 2, एक है तकरीबन 8.3% और दूसरा है तकरीबन 6.9%।
9.26.
6.92 है और 8.3। तो टोटल तकरीबन कोई 15% बनता है टोटल, 15% ऑफ द टोटल। तो ये वी थिंक दिस वाज एफिशिएंट टेक्निक फॉर यू टू यूज, बिकॉज यहाँ पे हार्डली ये सिर्फ 15% एक्सप्लेन कर रहा है आपकी जो पूरी उसको पिक्चर को। सो डोंट यू थिंक दैट यू हैव टू सम चेंज योर अप्रोच एनालाइज योर डेटा इन डिफरेंट मैनर, क्योंकि ये बहुत ज़्यादा एफिशिएंट मुझे नहीं लग रही आपको। यू कांट कंक्लूड ऑन बेस्ड ऑन दिस, ठीक है, क्योंकि आपने एक दफ़ा कहा कि गिलगित बल्तिस्तान में जो पॉपुलेशन डाइवर्सिटी है वो बहुत हाई थी, बाकी रीजन से खराब थी, लेकिन आपने साहब ने ये भी कहा कि जिसका एरिया वाइज कोई कोरिलेशन आपने वो भी फाइंड नहीं किया। तो ये दो चीज़ें कंट्राडिक्टरी हो जाती हैं कि हाउ कैन यू एक्सप्लेन दिस?
Sir, Kasortu Korikanchi, the Kada Munga principal coordinate analysis group, both the populations are overlapping, but there is a slight shift between the two populations. But that slight shift is not as clear that we can divide it into two populations.
I agree. मैं ज़रूरी खबर नहीं दी कौन वो ज़्यादा मुझे that percentage explain that it's only 15% of the total. So this is not very efficient in my view.
So actually the problem is when you have this diverse population, if you are in plural you can have like 60-80% explanation by the PCA 1 and 2, and then you just put one graph PCA. But that's why you have done coordinate analysis, you have done phylogenetic tree, you have done so many kind of analysis because you are working in a diverse population. So first answer is that. दूसरा वो आपसे गिलगित बल्तिस्तान का कह रहे हैं, जब आप कह रहे हैं कि शिफ्ट नहीं है या स्ट्रक्चर नहीं है, you are just talking about the simultaneous. वो basically will pass the data. Because they can, if you compare the data, then there will be some shift. But within your study area, there is no shift.
एक लास्ट क्वेश्चन एक आपने वो जो दो कंप्यूटर में आपने उस कंपैरिजन की प्रीवियस पूरा अच्छा जो पाकिस्तान के अंदर मेरी ये रिकमेंडेशन है कि आप इसको वो करें एक्सप्लेन करें, इसको थोड़ा सा डिस्ट्रिक्ट वाइज वो करना है, मतलब फॉर एग्जांपल बाजूवाड़ा है तो उधर भी बाजूवाड़ा होना चाहिए, इधर मुनीर है तो उधर भी मुनीर
where is the middle population? You know, never here, it's just a specific, the reporter में जो आंध्रा वाला का बता रहे, वो उसमें सिर्फ हमने 2010 में किया था, but he has done more diverse. ये तिगार भी गया था, खैबर भी गया था, बारा भी गया था, so this is the real tribal population. उसमें जो है वो सिर्फ एक लोकेशन का
Yeah, there's something wrong here. ये farmer के लिए कोई research का कोई मतलब के लिए
Farmer fields with your research field.
ये इसमें क्वेश्चन मेरा ये कि मतलब इसमें आपके पास कोई इस तरह की कॉमन होस्ट है जो मतलब ये सारी रीजन्स में कॉमन था जिस पे आप प्रॉपर जो है सिविरिटी को जो है कंपेयर कर सकें उसके लिए
Yes,
क्योंकि अगर आप एक रीजन से दूसरे रीजन में जाएंगे तो एक रीजन में एक तरह की वीट वैराइटीज वो ग्रो कर रहे हैं दूसरे रीजन में दूसरी तरह से ग्रो कर रहे हैं तो ऑफ कोर्स तो उनका जो रिस्पांस होगा पैथोजन को पोटेंशियल होगा तो आप उसको प्रॉपर जो है मतलब उस तरह कंपेयर